So my name is Luke Stetzik. I am a senior scientist at Aiforia. Thank you for joining us here today for this webinar for Aiforia and Global Engage. And the purpose of today's talk is going to be to discuss how to use deep learning tools in the context of analyzing histopathology and Alzheimer's disease. Our speaker for today is going to be Dr. Valentina Perosa. She is a research fellow at Massachusetts General Hospital, Harvard Medical School. But before we get into her portion of the talk, I just want to talk a little bit about the platform that Dr. Perosa used in her project. And so Aiforia is a cloud-based software that kind of breaks down into two major groups. Either it is utilized in the context of research, so this preclinical sector that we have here in the center of the screen. This is something where you would be able to use it for any kind of really custom fit research question that you wanted to answer, where you need to utilize and harness the power of AI, but maybe you don't necessarily have a background in AI or machine learning. I know that was definitely my case. So just a little bit of background about me. I recently joined the company, but prior to that, I had started out as a customer of Aiforia for about 3 years. And so, as I sort of give this intro talk, I might offer that perspective a little bit. And then in addition to that preclinical research aspect, there's also a clinical diagnostic center. And really the goal of this portion of the Aiforia platform is to improve the workflow and streamline the use of AI in a clinical diagnostic setting as an assistant tool. And really, I think that speed and that efficiency is one of those things that really shines with Aiforia in that context, and really any context, is that you're going to really increase the amount of the speed and the consistency of your analysis, as with any automated method. But I think really one of the things that sets Aiforia apart is the flexibility in terms of being able to adapt and continue to grow the way that you're analyzing your data, along with however the lab or the project is growing. And then also there are a lot of other systems that utilize something similar. Something similar in terms of being able to perform an analysis, but you may find that a lot of those systems are a little bit clunky in terms of the user interface. And one of the advantages that Aiforia has is that it's really very streamlined in terms of the tools that you need to use in order to develop and create an AI model and perform analyses. So Aiforia is a Helsinki-based company that has spread to the US and really all over the world. And I think part of the reason that this product has really exploded in the last few years is that we have a really great cross-section of people who are both from the tech and medical and research fields. And because we really have all of that talent to draw on, we're able to offer a really well-rounded product to groups that are either anything from big pharma, startup biotech companies, and top-tier academic institutions. As I mentioned sort of a little bit ago, there's sort of this main breakdown of research and clinical. And one of the things that we sort of like to brag about in terms of our clinical approach are our CE-IVD mark, AI models, things like Ki-67, ER, PR, all those kinds of things. And really, the thing that I like most about the clinical diagnostic suite is just how streamlined it is, and especially for pathologists, where essentially, it doesn't have all of the bells and whistles that you may get with the research side of things, but it has everything you need and nothing that you don't in terms of distractions. You have your images, you load them in, you're able to run your models, you're able to get your analysis. It has tools that allow you to make on-the-fly adjustments to that analysis. Maybe you see something you don't completely agree with in terms of the performance of your model. And then it also has the ability to complete your reports all within one place. The other aspect, this AI for preclinical analysis, there's really sort of two ways that works. And the way that is most commonly utilized by researchers is that they want to learn how to use the software themselves so that they're able to build an AI model that will work for maybe 1 or 2 specific projects, but also can be adapted to later projects as the lab grows and maybe new funding comes in or that kind of thing. And then alternatively, if maybe there isn't that kind of bandwidth in the lab to get comfortable in a new platform with a new software, but you do have an analysis and you do want to be able to utilize the power of AI in terms of enriching your data set. And for that, what would happen is we have a conversation about what it is exactly you want to quantify within your images, and then we can develop a bespoke model that will perform that analysis and give you those results that you need. And then the final component is an education-based component. And this is something that's really useful if you have a course that is really images heavy. Think a pathology course, something where you have to be able to distribute materials to a large number of people in a way that in classical or conventional classroom settings is arguably a little bit clunky. You may have some kind of portal interface where students can download materials and review them on their own. But with Aiforia, what you're able to do is have all of those things organized however you want, specific to your course, and have it in a place that students can access and is available, and also is able to direct their attention to the aspects of the images that are really important for learning, very easily eliminating, not completely necessarily eliminating, but certainly doesn't require in any way the use of on-premises microscopes, being in a classroom, or printed materials, things like that, that can kind of make the learning experience a little less individualized for students. I mentioned this a little bit earlier, but Aiforia is a cloud-based platform. And so it has all the advantages of being cloud-based. The thing that I really liked about this is that, as a customer, when I tried to get into AI and using AI in my projects, the first thing that I did was, I went to these Nature papers, I went to these, papers that had some kind of, open source model that I could go to. And the thing that I always have the problem with is that you download these models that work really well in one context on one data set and try to apply them to your own. And invariably, it's this endless list of dependency errors. Or maybe you go to another commercially available product and it has some things that are useful in it, but the interface is so challenging that you really kind of don't end up working in it as much and using it as much or as easily as you had anticipated. And even in those systems, sometimes you have on-premises hardware dependencies that, in our system with Aiforia, you really don't have to worry about. Everything is on the cloud, everything is secure, and all that you really need is a laptop with a decent internet connection in order to be able to upload your images, train your models, and perform the analyses that you want for your project. Additionally, there are a couple of things about our AI system that I think set us apart from other systems. So a lot of systems offer some kind of semantic segmentation, which is sort of this classical AI quantification that you see in the bottom left of the screen here. But anyone who has used one of these systems to try and quantify a more detailed measure, and really in the context of something like Alzheimer's disease, where you need to get counts of cells that are really close together, this is a classic limitation of semantic segmentation in that it will merge those two cells together. And with the system that we have tools that make it so that it's really easy to get very accurate counts of really heterogeneous populations of cells using object detection. And then additionally, if you have some more complex features related to those cells, say you want to quantify the activation state of glial cells or just any change in cellular morphology, the limitation within our platform is really the resolution of the images. You can add an object detection layer that will make it so you can quantify each cells and add instance segmentation to that object detection layer. And what that allows you to do is allow you to get the morphological patterns or morphological features of that cell, even if there's another cell right next to it. Because that first step is object detection, it will identify which portions of the image belong to that particular object, and then it'll segment those. So even if they are next to each other, even if you have processes that are overlapping, it really is able to sort of disentangle those things in a way that is honestly pretty impressive. And then the final component that I really like about Aiforia is that it's really, when you start off with a create platform as a researcher, we put you in contact with somebody, a field application scientist who can help guide you through the whole process. But for asynchronous learners, sometimes it makes more sense to have a big range of available resources. And within the community platform, there is everything that you need to know in order to be able to build and develop your own AI models. Without ever really having to, interface with somebody, or you can always come back and reference them. And even if you do have a more personal question, or you just want to get a feeling for how other people are, working within the platform, you're not probably the only one working on a very individual specific question, something general like, oh, I'm working on this, object detection. I'm trying to detect TH cells. Has anyone else worked on this? You can put that kind of question out into the platform, and you can see other people will see it, other people will respond. It's just a great interactive way to improve the model and the experience overall. And so with that, I will turn it over to Dr. Perosa.
Thank you so much, Lucas. Let me just share my screen. Can everyone see my slides? Lucas, can you see them?
I can see them.
Okay,
Yep.
perfect. Well, thank you so much again for the kind introduction. As Lucas said, my name is Valentina Perosa, and I am a research fellow at MGH and the Harvard Medical School in Boston. And today I'm going to talk a little bit about how we used basically Aiforia to investigate deep learning, to investigate methods to assess histopathological markers of Alzheimer's disease and cerebral amyloid angiopathy. So I am part of the Van Beluve lab, and we focus mostly on Alzheimer's disease and cerebral amyloid angiopathy. So therefore, I will give a little introduction about these diseases. And then I will dive into the methods that we adopt for most of the human studies in our lab, which are mostly ex vivo MRI, and what is more relevant for us today, the AI-derived measures of histopathology. Finally, I will also focus a little bit on what has been done in one of my main projects in the lab, which concentrated on the histopathological correlates of enlarged perivascular spaces. And finally, I will close, if we still have time, mentioning some other projects that are going on in the lab and also use AI. So first of all, what is cerebral amyloid angiopathy? It's a form of sporadic cerebral small vessel disease. And CAA is defined especially by the accumulation of amyloid beta in the small vessels of the brain. And this is especially in the perforating arteries that dive from the leptomeningeal into the cortex and then proceed into the centrum semivalve and the white matter. And this distinguishes a CAA from other forms of cerebral small vessel disease, which affect mostly the deeper brain arteries. So CAA can lead to dementia, but also to transient focal neurological episodes, to gait impairment, and most importantly, it can lead to devastating intracerebral hemorrhages, which often are lethal. For the diagnosis during life, we rely on typical neuroimaging markers of small vessel disease. And these can be seen on MRI and are, for instance, cortical superficial siderosis, but also microbleeds, so little microhemorrhages that you can see in T2 star weighted MRI, and intracerebral hemorrhages, as I just mentioned. But there are also other markers, such as enlarged perivascular spaces, which are represented on T2 weighted MRI as thin hyperintense stripes, and also white matter hyperintensities, which can be seen in the flare. So in particular, the hemorrhagic markers are crucial to determine CAA during life. And although our lab focuses specifically actually on CAA, it is very hard to talk about CAA without talking about Alzheimer's disease. And you probably know this, but AD is the most common form of neurodegenerative disease, and it leads to dementia. And its neuropathological correlates. So the neuropathological findings that we have in Alzheimer's disease are, for instance, amyloid beta plaques in the parenchyma, but also neurodegeneration and the accumulation of tau tangles. And although these are, by definition, the markers for Alzheimer's disease in histopathology, what we know from epidemiological studies is that also cerebral amyloid angiopathy is very widespread in Alzheimer's disease. Actually, we have studies that say that it's present in up to 89% of Alzheimer's disease cases. And in this respect, I always love to tell one little story, which is that the importance of CAA in Alzheimer's disease is not a new fact at all. And in fact, it has been it was already stated by Gaetano Perusini. And this guy actually funnily comes from my hometown, Udine, which is a very nice place in the northeastern part of Italy. And he already pointed out, as a collaborator of Alois Alzheimer, that cerebral amyloid angiopathy probably has a very big role in Alzheimer's disease. But unfortunately, this gentleman decided to join World War I, sorry, and he died on the battlefield as a doctor. So his family turned into producing wine, and nothing was heard about the CAA for quite a long time afterwards. So in the lab, we think actually that combining neuroimaging and neuropathology, so going from in vivo studies to ex vivo neuroimaging and finally to histopathology, is a very effective way to give some answers to pathophysiological questions and mechanisms that hide behind intracerebral hemorrhage, secondary tissue damage after bleeding, vascular amyloid beta deposition, and perivascular clearance. So a brain donation program was established at MGH with a focus on cerebral amyloid angiopathy patients, but it was then extended also to other forms of cerebral small vessel disease. So what happens is when a patient or the family of the patient decide to donate the brain and participate in the program, one hemisphere undergoes standard neuropathological examination, whereas the other hemisphere is fixed in formalin and then placed in a bag, which is then sealed and scanned at 3 Tesla MRI with a long protocol overnight in order to achieve a very high resolution for the scan. And the protocol includes a 2-2 weighted turbospin echo, but also a flash sequence and a diffusion weighted sequence. And after scanning, what we do is that the hemisphere is cut in 1 centimeter thick coronal slabs, and then samples are taken from predefined areas of the brain. So from the frontal, parietal, occipital lobe, and temporal lobe. But recently, we also started to look into the entorhinal cortex and into the hippocampus. And then the samples are embedded in paraffin and cut in 6 micrometer thick sections at the microtome. They are then stained for any marker that we wish to look at, or we perform immunohistochemistry for that. And through this cohort, it's possible to look into the histopathological correlates of the MRI markers of cerebral small vessel disease. So this is what is interesting about the cohort. And when we want to analyze a specific lesion more in depth, what we do is to first of all go back into the in vivo MRI, so the MRI that was performed during the life of the patient. Then we look at the ex vivo MRI, where we have a higher resolution and we can better locate the lesion that we want to look at. In this case, for instance, it's a microhemorrhage that you can see in which is a hypointense, so black in the cortical ribbon. And then we find back this lab of, in the pathology where this lesion is probably located. And then we also perform neuroimaging again at 7 Tesla MRI so that we have an even higher resolution and an even better localization of the lesion that we want to look at. We then find the piece of tissue where the lesion is located, embedded in paraffin, and then perform serial sectioning to find, for instance, in this case, the microbleed. So this one was not supposed to be there. And one of the things that we did more recently in the lab, as I was mentioning, was also to take this work a step further and to start using artificial intelligence, and in particular, Aiforia, in order to quantify histopathological markers of AD and CAA. And as you probably know, most of the times studies use semi-quantitative scales in pathology in order to determine a particular marker. For instance, we can talk about absent, mild, moderate, and severe CAA. But and sometimes even alternatively, manual count is used to determine the number of cells. So these are all things that Lucas just mentioned before. But one of the issues is obviously that by doing this, we have often subjective instead of objective markers, and it's very time consuming, and it's very hard also to bring this work a step further and maybe involve more centers into the studies. On the other hand, there are also semi-automatic assessment methods, such as the use of ImageJ, but this can be also susceptible to variability of fixations, slice thickness, the stain intensity, and so further and so forth. On the other hand, the use of deep learning, as we already mentioned, is objective. It gives us continuous measures, which are good because also it allows us to have better statistical models to investigate whatever it is that we want to investigate. It's time saving and it's easier to apply to several centers. So our lab has started to work with Aiforia, and with this tool, we create convolutional neural networks able to identify and quantify histopathological markers of AD and CAA, which have many common markers. The workflow for each model that we have trained consists in, first of all, obviously, the sample preparation. We then digitized the histopathological sections using a very high resolution digital microscope called the nanozoomer. The sections are then uploaded on the Aiforia cloud, and then we take 10% of the whole cohort that we want to analyze and perform annotations manually in order to train a model to identify the marker that we want to identify. After that, the model is applied when it's satisfactory to the whole data set. And finally, to make sure that the model really detects what we want to see, there is an external validation process that can also be performed within the Aiforia platform, for which we then annotate another 10% of the sections, or better to say, verify that the annotations made by the model is correct. When we are happy with all of this, we download the data and we can perform statistical analysis. So this is just one example of the step 1 and 2, so the sample preparation. Here you can see one histopathological section stained for amyloid beta. And Aiforia learns from, as I was mentioning, from labeled input data. And this labeled data represents the ground truth. Also, it's organized in layers. So for instance, here you can observe how in green, we first of all tell the model what is tissue. Also, we tell the model what is leptomeningeal tissue, which is important for CAA because the amyloid beta in CAA accumulates especially in the leptomeningeal and in the cortical arteries. So we want to tell the model, oh, this is leptomeningeal tissue, which represents another category compared to the other tissue. And within these layers, we have sub-layers in which we train the model to recognize CAA and distinguish it from the amyloid beta plaques that you can see in blue. So the result is that we can then, for example, determine the cerebral amyloid angiopathy percentage area. And since the model has also been trained to distinguish between tissue and the leptomeningeal vessels, we can also distinguish between cortical CAA and leptomeningeal CAA, which can be imported for the analysis. And secondly, we can determine the area occupied by amyloid beta plaques, which is definitely not, cannot be taken for granted because the color that we use to mark amyloid beta is obviously the same. So the models are then applied to the whole pathological cohort and validated, and the measures that can be used for this are precision, sensitivity, and F1 score, which is basically a combination of the two above. So here is an example of how we did that for the amyloid beta model, and you can see that the model performs very well also after the validation. So we repeated all these steps in order to build 6 different models. The first one was the amyloid beta model, but we also looked into fibrin, and especially fibrin within the vessels. And this can be a measure of a blood-brain barrier leakage, which is also an important pathophysiological process in cerebral small vessel disease. We then also look at the density of GFAP positive cells, in other words, activated astrocytes, and in the CD68 positive cells for activated microglia. We also detected the calcifications with another model, and finally also iron positive cells, which are very important for CAA because they detect hemorrhagic products and macrophages, which have taken over iron during the process. So once the models were performing well and had been validated, we also adopted them for statistical analysis. And first of all, the association between the measures obtained through the deep learning models and previously obtained measures through semi-quantitative scores was calculated in a subset of the CAA patients. And we could see how CAA, amyloid beta plaques, and also iron positive cells are very well correlated with the semi-quantitative measures. Also, in the same subset, we used a linear mixed effect model to look into the association between CAA and density of iron positive cells, which was positive and significant for leptomeningeal CAA. And this is important because it replicates something that we previously found in another study, which used the semi-quantitative score. So we could see that our measures and also the results of the analysis performed with the measures was consistent with previous studies from our same lab. So one project in which having these measures has proven to be particularly advantageous was that we performed on the histopathological correlates of MRI visible perivascular spaces in CAA. So perivascular spaces have been actually identified 150 years ago already, and we know many things about them. So we know that there are fluid-filled spaces that surround the cerebral small vessels, especially the smaller ones. We also suppose that they are visible in MRI as thin hyperintense stripes when enlarged, as I was saying before, on the T2 weighted MRI. And also, we know that the EPVS in the centrum semiovale are a marker of cerebral amyloid angiopathy, and in fact, they have been now considered as part of the criteria, of the new criteria for diagnosing this disease. Also, what is really important in this case is that they probably play a role in brain clearance pathways. And I don't know how many of you are familiar with brain clearance, but it's the process that allows the removal of certain toxic proteins from the brain. And it's very fascinating because it seems to have a big role in the development of Alzheimer's disease, or better to say, a failure of the system seems to have a big role in the building of Alzheimer's disease pathology. So when we started this project, there were many unanswered questions about enlarged perivascular spaces. First of all, what is the histopathological substrate of what we see in the MRI, so of the MRI visible perivascular spaces? And is the PVS enlargement that we see on MRI actually positively associated with the cerebral amyloid angiopathy pathology? Because as we remember, they are an important marker of this disease. And do the perforating cortical vessels that dive from the leptomeningeal into the cortex and then probably proceed into the white matter, do they relate with the enlarged perivascular spaces that we see? And are the perivascular spaces that we observe periarteriolar, perivenular, or both? So do they enlarge around arteries, around veins, or around both? And this is also important because it can give us some answers and also some hints about the actual routes of perivascular clearance, because these are largely still debated, not unknown, but debated. There are several theories about them. So in order to try to answer these questions with the study, we included 19 cases from the CAA cohort that I just presented and 5 non-CAA control cases. As already said, the intact hemisphere was scanned at 3 Tesla MRI, and the sequence that we took into account for this was the T2 weighted sequence to look into perivascular spaces. So for the histopathology, we took the 5 predefined areas for histopathological analysis. We took the cortical ones from temporal, frontal, parietal, and occipital lobe, but also one section from the basal ganglia. And the first aim was to demonstrate the relationship between MRI visible perivascular spaces on in vivo MRI and post-mortem perivascular spaces enlargement. So between the MRI scanned during life and what we scanned after. And we could see in this case also that the severity of the enlargement on in vivo MRI very well correlated with the severity of the enlargement on ex vivo MRI. And this can sound very trivial, but had not been done before. And then the second step, we looked at the histopathology, and in particular at sections stained for Luxol blue and HD, because they provide a very good contrast between the space itself and the parenchyma, or the white matter. And you can see in this enlargement, for example, one example of a perivascular space, which is located around the vessel that you can see in the middle. And in order to prove this, what we did was we went back to the ex vivo MRI, and we established the severity of the MRI according to the severity of the enlargement of the perivascular space according to the region that was then sampled. So we used for that a semi-quantitative score, but then on the histopathological sections, we automatically segmented the enlarged perivascular spaces using an in-house developed tool. And what we could see after correlating the ex vivo MRI with the histopathology is also that in this case, we saw that the regional severity of PVS enlargement on ex vivo MRI was positively associated with the percentage of EPVS area on the corresponding histopathological samples. And this was reassuring because it told us that we were looking at the right thing, especially because there has been some debate about whether what we see in the histopathology is possibly an artifact and not really the enlarged perivascular space. And also there was a significant difference between the severity of perivascular spaces in the CAA compared to the non-CAA control cases, but this was not very surprising. And the further aim was to look at the association between percentage area of enlarged perivascular spaces and a measure of CAA. And we did this in the cortical regions of the CAA subjects. And the percentage area of the enlarged perivascular spaces was derived from the LHE sections using the semi-automatic method that I've just mentioned. And what we saw was by using a linear mixed effect models, setting subjects and cortical region as random variable and always sex and age and age at death as fixed factor, that the total percentage area of a CAA was related to the percentage area of the enlarged perivascular spaces on histopathology. And this is interesting, like the fact that we could use a linear mixed effect model was only because we had the continuous measures of histopathology obtained through the deep learning. So to this model, we also had a further fixed factor that we thought could be associated with the severity of the perivascular spaces. One obvious cofactor could be amyloid beta plaques, but we found no relationship with the enlarged perivascular spaces. We found a relationship with myelin rarefaction, and also no relationship to fibrin-positive vessels, which, as a reminder, were a marker of blood-brain barrier leakage. So what does that tell us? How do we explain this association? So in CAA, amyloid beta actually accumulates in the cortical vessels, and just very rarely we can see any CAA at all in the white matter. However, the enlarged perivascular spaces that we observe are actually located in the white matter. So one possible explanation for this relationship is that clearance impairment in the cortical vessels due to amyloid beta accumulation leads to a clearance block and therefore to an enlargement of the perivascular spaces in the downstream portion of the same vessel. But before jumping to this conclusion and before being able to really verify this, it was important to look into the anatomy of the enlarged perivascular spaces and most importantly, into the anatomy of their related vessel. So the question was, is really the same vessel that goes through the cortex and accumulates CAA, also this vessel around which the perivascular space enlarge? And in order to do so, in order to answer this question, we individuated 2 CAA cases, one with severe and one with moderate CAA. And based on the in vivo MRI, we selected a parieto-occipital region, and we cut a block out of the formalin fixed tissue. And then we placed this block in a falcon tube. First of all, we also placed it in PBS in order to restore scanning parameters. After that, we placed it in Fomblin to avoid MRI artifacts. We removed air bubbles very carefully because they really undermined the quality of the scan. And then we placed this falcon tube into a custom-made solenoid coil. The scan was lasted then for 24 hours and included a TEC sequence with 100 micrometer isotropic resolution and the flash sequence with a 75 micrometer isotropic resolution, which is very high. You can see here from the co-registration between this scan at 7 Tesla and the in vivo and the ex vivo 3 Tesla scan, where this little piece of brain was located. And in this video, you can also see the result with the average TEC sequences. You can see very nicely the hyperintense EPVS in the white matter, and in some cases, also the related vessel within the enlarged perivascular space, which is, in this case, hypointense. And the first observation that we can make in most cases is that the perforating arteries, as we suspected, the ones that dive into the cortex also continue into the white matter, and that the enlarged perivascular spaces are around these vessels. So in the enlarged perivascular spaces, they also continue sometimes into the gray matter, as you can maybe observe. So what seems completely disconnected on 3 Tesla MRI is obviously has a continuation between cortex and white matter at 7 Tesla MRI. And after scanning, both blocks were then cut and placed into thin cassettes. They were then cut into serious sections, and these sections were alternatively stained for H&E, but also amyloid beta and smooth muscle cells. And we could confirm the observation made initially at the 7 Tesla MRI, which is that sometimes the enlarged perivascular space continue in the gray matter, as you can see in B1. And furthermore, we observed that the enlarged perivascular spaces related vessel have a dominant positive stain for smooth muscle cells, which is indicative of the fact that we are looking at arterioles and not at venules. So here you can see another example of an EPVS related vessel in C1, which stains positively for smooth muscle cells, whereas around the vein in C2, you don't see the enlargement of the perivascular space. So this is also consistent with some work done at high resolution in vivo MRI. And in the serious sections of the severe CAA cases, we sometimes also found some CAA in the white matter, not much, but a little bit. And we could see that, at least visually, that this CAA was found especially in vessels in which the perivascular space was enlarged, as you can observe in E1. And we thought that this could be suggestive, again, of clearance impairment in the upstream portion of the vessel, as if the amyloid kind of remains stuck in the attempt of being transported in the upstream direction. And when we tried to quantify this, we could also see a relationship between CAA in the white matter and the enlarged perivascular spaces area. So what we think is that the amyloid could remain stuck in the vessel wall, a little bit like the trucks on their way to the UK during Brexit, or after Brexit, better say. So hopefully this work can suggest some answers to the questions that we asked. So the degree of MRI visible perivascular spaces in the juxtacortical brain areas very well reflects enlargement on histopathology. Also, perivascular space enlargement is associated with increased CAE accumulation in the overlying cortex. And the perforating cortical vessels relate to the enlarged perivascular spaces in the juxtacortical white matter. And what we observe is that the perivascular spaces mainly enlarge around arterioles and less around the venules. And what this suggests for brain clearance is that clearance probably happens alongside arteries and towards the brain surface. At least this is what we think in our lab. So I would just like to jump to a completely different topic, actually, which is about cortical superficial siderosis, which is another marker of cerebral amyloid angiopathy. And you can see this in the MRI scan on the upper left corner. And this is work done by one of our technicians, a very young, talented Corinne Auger. So she looked into cortical superficial siderosis because this also recently emerged as a very strong predictor of subsequent intracellular hemorrhage. So when patients have cortical superficial siderosis, the possibility that they also get hemorrhage is much higher. And what we did, what we wanted to look at in this project was the association between cortical superficial siderosis and inflammatory reactions in the parenchyma. So what we did was, first of all, a double stain to see if reactive astrocytes, which are indicated by GFAP positive cells, co-localize with iron on a cellular level. And we found that they did, which kind of suggests phagocytosis. What we did then was to use the output from Aiforia to make maps of objects' locations. So here, each iron deposit is represented by a black dot, as you can see in the middle, and we can look at whether the area that we want, and get the density of iron from the fraction of the pixels that in that area, which are black. So we then co-registered the GFAP stained sections with the iron stained sections in order to determine the overlap between these two markers. And what we found was that there was a local density of reactive astrocytes that was strongly associated with the local iron density, suggesting a neuroinflammatory process. By using these pixels that we then defined in the heat maps produced by Aiforia, we could also see that most of the effect of iron deposits on the reactive astrocyte density is observed within the first 500 to 1,000 micrometers distance from the cortical superficial siderosis. And all of this basically tells us that neuroinflammation probably has a role in cortical superficial siderosis, although we did not find the same effect for CD68 positive cells. So I would like to thank you very much for your attention. And this one is the Van Verluve lab in which I work and who mainly works with this deep learning derived data. Thank you so much. Happy to take any questions, of course.
Yes, thank you so much. Looks like maybe we've got one question here. It says, have you considered extending your histopathology workflow to include a tissue microdissection step to generate genomic data for the ROIs?
Yes, that's a very good question. So we did consider that, and we are actually in the process of implementing that as well. But to be honest, by looking more into RNA than into the genome in itself.
Nice. I had some questions sort of about your main findings. So you did a really, I think, a really great job of showing that the perivascular, enlarged perivascular space correlates with the amyloid deposits that you were showing. I don't want to put you too much on the spot, but like sort of anecdotally, why do you think, that correlation exists? And obviously it's important, but yeah.
Well, as I was saying during the presentation, I think what we think is that it relates to the impairment in clearance pathways. So CAA has been related already to clearance pathways, but also the enlargement of perivascular spaces has been suggested by many as a possible marker of dysfunctional clearance, which means that what we think, as I was saying, is that the amyloid remains stuck in the vessel walls, and that's where we have this correlation between the amount of vascular amyloid and the enlargement of the perivascular spaces in the downstream portion of the same vessel. So this is also why perivascular spaces in general have gained a lot of attention in the community recently.
I guess what I should have said was more, does it seem pretty clear that there is, this connection? So there's obviously a correlation, but is the perivascular space enlarging, as a product of those things accumulating, or are those things accumulating because that space is enlarged? Obviously.
Yeah, I mean, obviously, this would require a functional study that we cannot perform in humans. But I think what is kind of exciting in this case is that we were able to show that the vessel around which the space enlarged are the same vessels around where the amyloid beta remains stuck. So I think that's kind of also the interesting connection that we make here, which is obviously not a causal connection, because for that we would probably need mice experiments, and there are plenty of mice experiments out there, but we are interested in human studies. So we can, to answer that question, what probably would be needed is sort of a cohort with many different stages of CAA, and that could probably at least partially answer the question of sequentiality.
Awesome. And so then I guess just two other quick questions. So you showed a graph that had your automated data quantified versus the scoring. And for the most part at the very beginning, and at the most part, the two correlated really nicely. But I did notice that there were some outlier cases. And so I guess my question is, was there anything really special about those outlier cases that you noticed? Was it a model performance, or was it, maybe? Go ahead. Yeah, I think you get the question.
No, that's a very good question, actually, which we tried to answer a little bit, at least in the related publications. So what we found was actually that both of them, sometimes the model would a little bit fail, but it was not necessarily the model which would fail, but maybe for the iron, for instance, there were some blue spots which were there due to the staining process, and that would be picked up as iron deposits, obviously, by the model. So that's like a staining mistake. The other thing that we sometimes observed is actually that the human eye, more than the AI, misunderstood the severity of a certain marker. So that was it was very interesting and reassuring. But of course, I would still underline the importance of performing a visual quality control for also for the AI models, because without it, it's still, it's just necessary as a backup.
Awesome. And were there specific questions that were not necessarily part of your initial aim, but as you collected data and started to see it? So you just mentioned that there were some instances where it seemed like the model was performing really well, but wasn't consistent with the way that people had assessed things visually. And I find that in those situations, that's usually sort of where you sort of see new things, and then it kind of gets your gears turning about. And so I was just wondering if you had any other directions you were planning on following up that you could share that, kind of came out of that process.
I don't think that's that my new, let's say, our new ideas are not necessarily driven by mistakes made by the human eye or by the performance of the model. But I think what is extremely exciting is the opportunity to have these heat maps produced by the models. So that really sparkled a whole bunch and range of new possibilities. For instance, what I would really love to do and next is to relate the pathology of the vessel, so then directly with the perivascular amount of certain pathologies. So that's something for which it's great to have these heat maps, because it allows to co-register them and look into the spatial relationship between several markers. So I think that's fantastic.
We do have one other question that showed up here. Would the imaging be robust enough to ask the question, is the astrocyte foot processes decreasing around vessels affected by amyloid?
If so, the astrocyte processes are affected by amyloid in the vessels.
Right. So
yeah, I'm not sure whether, I mean, we do see visually an increase in the density of reactive astrocytes around the vessels with amyloid, but I'm not sure that the image itself is enough to really look into the astrocyte processes. It's like actually fantastic idea though. I should go back to the sections and look better into those.
Nice. All right, well, I think that's all the questions that we have. I'm not sure if there's anyone else who's got anything. If not, thank you so much for sharing your work with us. That was a really great talk. Yes, and enjoy the rest of your day. Thank you everyone for being here. Yeah.
Goodbye. Bye-bye.
Bye.